Have you ever thought about why pirates are always drawn or depicted with eye patches? You might be thinking, well, they all have injuries to their eyes or they're missing an eye, and that would be weird if all pirates had the same injury to the same eye. But in fact, the eye patch helped them adapt to darkness really quickly. You see, there was no electricity, so when you're on a boat and you're in battle or you have to go below deck, you have to be able to adjust to darkness. So by covering the eye was able to quickly adjust to darkness and they could lift it up, find what they're looking for, go to battle, or do whatever they wanted. Now this video is not about pirates, but it is about how cool the eye can be. So let's talk about vision. Alright guys, welcome to Psych Explained. In this video, we're going to examine the visual pathway from start to finish. How information is first received at the eye, and we'll go over those parts, all the way until it's interpreted and processed in the brain. Now as you look around you see tons of objects. You might see faces like mine and colors like my purple shirt or the flower. And it might seem like your eye recognizes faces and recognizes colors. But all your eye is doing is taking in raw data that's reflected off of the object. And this raw data comes in the form of light waves or visible light. You see, there's energies and wavelengths all around you that you just don't notice or can't perceive, like microwaves and X-rays and gamma rays and radio waves, right? But our eye has evolved to detect a specific type of wave or visible light. And the colors we see depends on the wavelength. We usually talk about long, medium, and short. For example, long wavelengths, okay, typically like this, help us see the color red, and wavelengths are usually determined by peak to peak. Or something like medium wavelengths, okay, help us see the color green, okay, something like this. Or we have short wavelengths that help us see things like the color blue. So this is kind of a nice intro in terms of what we're actually perceiving, right? All of our senses, our nose, our ears, take in raw data and then we interpret it later on. All right, so let's start here. Let's start with light. Light is going to reflect off this flower, okay? Where does it go? Into our eye. And the question is, what is it going to hit first? Well, the first part it's going to hit is this outermost layer here, and I'm going to kind of shade in so we can see it, is what we call the cornea. Okay, here's the cornea. You might be thinking, what is the cornea? Well, first, we'll use a different marker here. This is considered the outermost layer. Okay, the outermost transparent layer. And whenever I hear about on TV the athletes getting their eye scratched, I think of a cornea abrasion, something happening to their eye. In addition to the outermost layer, it helps bend the light and bring the light into the eye. And where is that light going to go next? Well, that light is going to go through this dark little black hole. What do we call that? That is your pupil. Okay, right here is our pupil. And what does our pupil do? Our pupil, well, essentially, as I said before, is our black hole in the middle of the eye that takes in the light. Now here's a question. What determines how much light enters our eye, right? If you're in a very sunny place or very dark place, that's going to dictate how much light enters your eye. And what determines? This little structure, this muscle that is brown, this is what we call the iris. Okay, so this person has a brown eye. Okay, your iris is the color of your eye. So it's always funny whenever we talk about, this person has beautiful eyes. What you're really saying is they have a beautiful iris. I would not recommend that for a Valentine's Day card, but that's what we are saying. So what is your iris? It is the muscle, okay, that controls the light that enters the eye. Okay? Now, there's a couple words I want us to know. There are terms like dilate, okay? You might think, what does dilate mean? This is when your eye, the pupil, gets really big, right? The iris is expanding. Now, when would that happen? That's when it's really dark, and we want a lot of light to enter our eye, okay? What's another one? We have constrict, the opposite. What is constrict? That's when the pupil closes. Or the iris closes, right? We don't want too much light in. That would be if it's very bright and sunny outside, and the iris is gonna go like that. Now, here's a bonus question. is not just about light that dictates how much light enters the pupil. It's also about your emotions, like love and fear. So, let's say you're scared, right? You're about to do some public speaking. What part of your nervous system is going to control how much light enters your eye? Because you know when you're nervous, your pupils are going to start dilating. What's the answer? Our autonomic nervous system, or more specifically, our sympathetic nervous system, right? This is our fight or flight. So it's not just light, it can also be our emotions. So we have light entering through the cornea, the outermost layer, through the dark hole called the pupil, which is controlled by the iris, which is a muscle. What happens there? This light is going to be reflected in this little structure right here. It looks like kind of a football. This is a really important structure. What is the structure? This is the lens, okay? Right here, okay? Now, what does the lens do? Well, do you ever see somebody with a big professional camera and you see they're always adjusting like this? They're adjusting their lens, to have focus. And that is what our lens does. It helps focus the light, focus the image onto the back layer of the eye, right? So if something is coming towards you, your lens is constantly going to I feel like it's like a water balloon. It's constantly going in and out. Well, that's what your lens does. It's constantly changing shape depending on whether you look at something really far or really close. Now, how does your lens actually know whether something's far or close or determine distance? Well, in psychology, we talk about something called binocular cues, right? We use both eyes, right? We take information from both eyes and we interpret it in our brain to judge distance and depth. So that'll help us determine distance and how much we need to focus. Now, there are two binocular cues. One is called retinal disparity. The other one is called convergence. When something comes really close to you, right, you can see your eyes almost converging. Now, it's also important to know is that when your lens does change shape, there is a term for that. And we call this accommodation. What's it called? Accommodation, right? To accommodate, accommodation. Now, do not confuse this because Jean Piaget uses this term in developmental psychology. When you change your existing schema, when you approach a new experience, Piaget would call this accommodation, right? The opposite would be assimilation. So this refers to the eye anatomy, all right? Now, what does the lens do next? Well, the light is then going to be reflected, projected onto the back layer of the eye. So here's our light being projected on the back layer of the eye. Now, what is the back layer of the eye called? This is really, really important stuff. This is what we call, I'll write it right down here, the retina, okay? This is where all the magic happens. So our retina covers the entire back layer of the eye. And I'll just kind of point to this over here just to make sure we know it's on the whole back layer of the eye. Now, why is the retina so important? Remember, with all of our senses, you have to take some sort of energy like light waves and sound waves or odor molecules, and you have to turn that to electricity because that's how your brain makes sense of things. So this is where light waves, or visible light, converts into electrical signals. Now, do we know what that's called? We've talked about in other videos. When light converts into electricity, we call that transduction. Okay, transduction. This happens in all of our senses. So here we have light converting into, now we can use the word action potentials, neural impulses. We're going to say action potentials, right? Tons of words for it. APs, action potentials. Potentials. All right, now this is our retina. Now let's actually zoom in on the retina because there's a lot of really important stuff there, okay? This right here is a zoomed in version of our retina. So let's say we're like looking at a cross section right here, okay? So this becomes right here, okay? So what are we looking at? Well, here's the retina, the back layer, and let's write that in so we know we're looking at the retina. I'll write it on an angle as much as I can so you know, here's the retina, okay? So what's happening? Well, light has to hit the back of the eye, right? So light is gonna go through all these little cells, okay, towards the back of the eye, and it's going to hit this back layer right here, okay, the back of the eye, back of the retina, okay? So this is the photo light sensitive layer. Now there's a lot of cells in the retina, but there's 3 of them that we're going to focus on in the video. Photoreceptors, bipolar cells, and retinal ganglion cells. So let's take a look at them. We have what? We have photoreceptor cells, all these cones and rods. We'll dive into those. We have number 2, bipolar cells that they connect to. And we have lastly what? Ganglion cells, or retinal ganglion cells. Boom, boom, boom. And then eventually those will go to the brain. All right, let's start with number 1. Let's start with photoreceptors. Photoreceptors. Perfect. All right, so what are photoreceptors? These are the cells that convert light into electrical signals. What do we call that again? Transduction. All right, there are 2 types we need to know. The first, the little arrow here, is called cones. Okay, what are cones? Cones are all of the ones, all these little cells that are colorful. We have green, we have blue, we have red. These are what we call cones. Now, what do cones do? Well, cones, a few things. They mainly are sensitive and are detected during daylight. Okay, so they will fire an extra potential when it's very sunny outside. Okay, they're going to help us perceive color. Okay, and also fine detail, like when you're reading a book or reading a text message. Okay, now another name for fine detail, you might see this in a psychology textbook, is something called visual acuity. I know that's a big one, but I almost feel like when you're at an eye exam and you have to close one eye and look at and see, do you have 20/20 vision? That's what they're testing for, visual acuity, all the fine details you can see. Okay, now most importantly, okay, this is important, where can we find most of the cones? They're mostly concentrated in one area. Right? Where are they concentrated? Concentrated around the fovea. All right, what is the fovea? You see this little pit right here? That's the fovea. Okay, so in fact, let's just write fovea here, and we'll have a little arrow so we know what we're looking at. Okay, so there's the fovea. Okay, why is the fovea important? There's only cones located in the fovea, and the most amount of cones are located in the fovea. So yes, you have cones all around. The majority of them are located right there, which means we have the best, sharpest vision right in the center. Okay, so we can actually add, some C's here, so we know these are where our cones are primarily located, right in the fovea. All right, what else do we have? The second cell, opposite of cones, is what we call rods. All right, what do you know about rods? Well, it's kind of the opposite. Instead of daylight, rods help with night vision. Okay, so when you turn the lights off, and all of a sudden, you can't see. Well, your rods have to get accustomed and acclimated to it yet, right? It takes a little while for your rods to get activated. So it's not just night vision, but things that are black and white. That's really important. Also with rods, rather than concentrated on the fovea, these are concentrated in a different place. So where could they be? They are concentrated on our periphery. Right into there. So what is our periphery? Peripheral vision, right? Central vision, peripheral vision. So what does that tell us? It tells us that it's really hard to see colors in our peripheral vision and see fine details, right? Next time you're reading a book, reading a text message, right? I want you to focus on one single word. What you'll notice is that one word is very clear and you can see the details, but all the words around are kind of blurry. That's because rods don't help with fine detail. Only cones do. All right? So we have rods that help with night vision, black and white, mostly concentrated on our peripherals. So let's actually have a different color. If we know C is cones in the middle, we're going to have mostly rods on our peripheral. Now we do have cones on our peripheral vision, but maybe not as many. Okay, so we have rods, and mostly cones right in the center. All right? So that's our first cell, photoreceptors. Now what's next? We also have, I'll do number 2 here, bipolar cells. Bipolar cells, okay? And what do bipolar cells do? Well, they basically receive action potentials from the cones and rods. You see, it's kind of like a step-by-step process. We have light hitting the back of the retina, which activate the cones and rods, which then activate the bipolar cells, which then finally activate the retinal ganglion cells. Okay, it's a step-by-step process. Okay, now for me, the ganglion cells are more important. Why is that? Because the ganglion cells, the axons turn into the optic nerve. You see, what actually takes the action potentials from the eye to the brain? There has to be some place. Right here, the optic nerve, okay? So all of these little axons right here are gonna turn into the optic nerve. They're gonna go fire, 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 and it's all gonna go down the optic nerve right here, okay? So let's write in here, we have the optic nerve. Okay, so all of our action potentials are traveling down there. Now there is one thing to notice. There's one place we don't actually have cones and rods, okay? Where's that? Well, it is right here, okay? There's nothing there. Why is there nothing there? Because that's where the optic nerve leaves the eye. There is no retina, right? There is no place. So what do we call that? Well, this is the blind disc or the blind spot. Okay, so what does that mean? There are no cones and rods. Okay, so boom, boom, boom. Now, here's one memory technique I have for a few of these. How are we going to remember that cones help us see color? Well, CC. Cones, color, CC. Also, how do we remember the order? Photoreceptors, bipolar, ganglion. Well, this might sound bizarre, but I know that the letter B comes before the letter G. So bipolar, then ganglion. If it helps you, I hope so, but at least it helps me. All right, so there's the process of the eye. Now, what happens in the brain? All information has been received. The action potential is traveling down the optic nerve. Where are they going? Well, let's take a look here. We have our optic nerve. We have our eye, lens, all that good stuff. We're going to travel up here. Here's our action potentials traveling up the optic nerve, the optic tract. And where are they going to go? So what is this structure? Right here. This structure is what we call the thalamus. And I do have a separate video on the thalamus if you want to watch it that kind of dissects everything. But what we got to know about the thalamus is the pit stop. All of our senses, except for smell, stop at the thalamus. And the thalamus is like, where should we go next? Right, it's the relay center. It's going to decide where things should go. Now, specifically, within the thalamus, okay, is a bunch of nuclei, a bunch of neurons, called the lateral geniculate nucleus, okay, which is specifically for vision. So the action potentials travel up to the thalamus, the LGN, and the LGN says, where should we put this message from the eye? I know where we should put it. Let's put it where we process vision. So where's that going to go? The back of the brain, all the way here. What is this called? The primary visual cortex. Let's write that in together. The primary visual cortex. And you might see in some textbooks, they say the V1, right? And there's V2, V3, V4, all the way. There's tons of visual cortex, right? This is where interpretation happens. Now, bonus question. What lobe will we find the primary visual cortex? Let's practice. We have our frontal lobe, our temporal lobes on the side. We can remember that because they're by our temples. Our parietal lobe receives information about touch and temperature from our body. Lastly is our occipital lobe. So where is this? This is our occipital lobe. Okay, all the way back here. And this is what's going to help us interpret all the information. Now, once it reaches the occipital lobe, our occipital lobe doesn't really know what it's looking at. In other words, things have to happen. So for example, information might travel up this stream right here. This is often considered the stream of like spatial orientation, like how far is something from me? What am I looking at? This stream is going to go to our temporal lobe. Okay, this is kind of the what. What am I looking at? Object identification. Right? In other words, once you see something, then we have to match it to our memories. And have we seen this before and things like that. Now, last thing I'll say before I get to this one is there are actually cells within your visual cortex that help us make sense of things. You see, if I'm seeing a flower, my visual cortex does kind of help me know what I'm looking at. These are called feature detectors. Okay? Why do we call feature detectors? Because they help us detect the features of objects. Things like, lines and shapes and angles, right? So it's almost like when you're looking at the potted plant, it's almost like making it out for you, right? You can't really make it out, but it is kind of showing you what we're looking at. I'm trying to draw my potted plant, right? This is kind of what our features detectors will do, right? And they'll send this message to other parts of the brain and be like, oh, that's a flower, right? And think about how instantaneous this happens. All right, so now that we know our primary visual cortex, What is this showing? Well, it's important to know that everything's kind of crisscrossed. I know it's kind of weird to think about. But what happens is everything on my left side of my body is processed in my right hemisphere, and everything on my right side is processed by my left hemisphere. We often call this contralateral control, contra meaning opposite. So here's just a nice example. If I see something in my way left visual field, like a friend or something, that's going to be processed towards the inside of my eye, towards my nose, and that information is going to travel through my optic nerve, down to my LGN, my thalamus, and then make its way to the opposite hemisphere. So left side to my right hemisphere, okay? All right, what about the other side? Well, on my other side, let's say I'm looking at something in my far right vision. Well, that's going to be processed right here, close to my nose, and that information is going to travel down the optic nerve, right, the action potentials, and it's going to crisscross down the LGN and then to my primary visual cortex, okay? Now here's what's interesting. You notice that they cross, okay? Why is that important? Because there's a name for it, and it's important to know that this is where all the action potentials cross from both eyes. Do we know what that's called? It's called the optic chiasm, okay? Optic chiasm. So this is where, pull X here, your optic nerves cross, okay? All right, guys, thanks for watching. I really hope you took something away. Don't forget to like the video, subscribe. I'll see you next time.